This method can help answer questions such as how many ribonucleotides are incorporated into nascent, leading, and lagging strand DNA in the genome.
Though this method can provide quantitative and qualitative information regarding ribonucleotide density in the yeast genome, it can also be applied to other systems such as mammalian genomes.
The alkaline gel electrophoresis provides a semi-quantitative view of ribonucleotide incorporation in the whole genome, and strand-specific southern blotting allows for greater sensitivity and quantitation at a specific genomic locus.
To begin this procedure, add 6 microliters of 1 molar potassium hydroxide to each sample of freshly prepared yeast genomic DNA and incubate at 55 degrees Celsius for 2 hours. After 2 hours, incubate the samples on ice for 5 minutes. Then add to each sample 4 microliters of 6,6-alkaline DNA loading buffer. Cast an alkaline gel comprised of 1% agarose, 50 millimolar sodium hydroxide, and 1 millimolar EDTA pH 8 with a comb that allows for 24 microliters of DNA sample to be loaded per lane. Spin down the tubes of samples briefly. Load the entire 24 microliters of each sample into the alkaline agarose gel. Include an appropriate DNA size marker. Electrophorese at 30 volts for 30 minutes. Then lower the voltage to 10 volts and electrophorese for 18 to 20 hours. On the following day, neutralize the gel. Immerse the gel in neutralization buffer.